We did paper number 4 on the first in-human trial of an RNAi therapeutic targeting VEGF and KSP in cancer patients with liver involvement. The hypothesis of the study was using LNP-formulated siRNAs is a safe and effective way to treat human disease involving cancer with liver involvement. The objectives of this paper are as follows: to harness RNAi to silence genes involved in disease, to provide evidence of LNP being effective in delivering siRNAs to humans in vivo, and to make sure that the LNP-formulated siRNAs is safe and well tolerated. Some terms to know and to keep in mind throughout the presentation and throughout the study is RNAi, which is RNA interference, and it's a mechanism for silencing gene expression. The mechanism is done in 2 steps. First one is degradation of double-strand RNA to siRNA with the help of an enzyme called dicer. The siRNA then binds to the RISC complex with the help of Argonaute proteins, and this mediates the target mRNA degradation and cleavage. siRNA, or short interfering RNA, is what's used to interfere with the translation of proteins by promoting the degradation of mRNA. LNP, or lipid nanoparticles, is a method of drug delivery, and this is what's used to deliver the siRNAs to the liver in this study. ALN-VSP is an LNP formulation containing 2 siRNAs that target the 2 target genes in this study, which is VEGF and KSP. This is the RNAi therapeutic that's used in this particular study. The 2 target genes in this experiment is VEGF, or vascular endothelial growth factor, and it's a key mediator of angiogenesis, or the formation of new blood cells. It's important to this study because of the role it plays in tumor vasculature, and this makes it a target for anti-cancer therapy. KSP, or kinesin spindle protein, plays a critical role in mitosis, more specifically in the proper separation of spindle pools. The inhibition of KSP leads to cell cycle arrest and apoptosis, so this makes it important to the study because it targets rapidly dividing cells. So a quick overview of the introduction of this study. RNAi is used to silence genes involved in disease through the development of a therapeutic called ALN-VSP. So since RNAi pathways are present in all mammalian cells, the primary challenge lies in the in vivo delivery to the appropriate organ with productive cellular uptake, which will lead to the siRNAs to bind to the RISC complex in the cytosol. The reason they used an in vivo approach was due to the benefits of using a living organism and to see how it's being affected biologically. So what they came up with was the LNP formulation. They had previously tested other nanoparticle formulations, but LNP had been proven to be the most effective in delivering the siRNAs to the liver and silencing other hepatocyte gene targets across multiple species, including rodents and primates. The results in these animal models proved to be successful and promising, so these findings were then translated into an effective way to deliver the siRNAs to human disease. So all in all, to examine the safety and effectiveness of this LNP formulation, a phase 1 trial was initiated to treat patients with advanced cancer and liver metastases, and that's going to be explained more in the next few slides.
I'm here to talk about the methods and procedures and materials used during this trial. First thing I'm talking about is animal experiments that had taken place. They used severe combined immune deficient SCID beige mice for this experiment. And one reason they use them is because they are B and T lymphocyte deficient, which it just caused them to display a major immune deficit, and they provided a unique biological setting that can be used to address major questions, especially during this type of experiment. And intrahepatic tumors were generated by injection of 1 × 10⁶ human Hep3B hepatocellular carcinoma cells. Cells were authenticated through analysis of protein expression, presence of Hep B viral DNA by PCR. The injection was into the liver of the skid beige mice, and tumor-bearing mice were given a 4-week treatment to determine the anti-tumor effects of ALN-KSP. So what they did to analyze all the data was RNA was purified from a blinded biopsy sample. Quality and quantity of RNA was assessed with the Bioanalyzer 2000, which is a chip-based capillary electrophoresis machine used to analyze RNA, DNA, and protein. It utilizes a micro-fabricated chip requiring minimum sample consumption and helps optimize PCR reactions for gene expression, sequencing, cloning, and typing. See, QRT PCR was conducted using a light cycle 480 Pro Master Mix. QRT PCR data analysis was conducted using the Relative Expression Analysis Tool, or REST. REST is a software design that compares 2 or more treatment groups or conditions with up to 100 data points in sample of the control group for multiple reference genes and up to 15 target genes. The mathematical model used for REST is based on the correction for exact PCR efficiencies and the mean crossing point deviation between sample groups and control groups. 5' race was conducted for either KIF11 or VEGF-A. Standard Illumina genomic sequencing adapters were added to the second round of 5' race PCR primers in sequence using custom Illum sequencing primer. Sequences were aligned to the appropriate transcript, and the cleavage portion in KF11 is 1249, and for VEGF-A, it's 139. Let's see, phase one. For the phase 1 clinical trials, the patients had to be 18 years or older with advanced solid tumors in the liver, and standard therapy were used in the trials. And no standard therapy were used in the trials. Other criteria were the aspartate and alanine aminotransferase levels were less than or equal to 2.5 times the upper limit, and aspartate and alanine aminotransferase are enzymes that the liver makes, and when the liver is acting normal, they are low in. With enzymes, when your liver is working normal, the levels of the aspartate and alanine are low, and when the liver isn't functioning properly, the levels increase. The total bilirubin is within normal limits. Albumin greater than 3.0 grams per deciliter. Absolute neutrophil count greater than or equal to 1,500 cells per millimeter cubed. Platelet count greater than or equal to 100,000 cells per millimeter cubed. And hemoglobin greater than or equal to 9 grams per deciliter. Serum creatinine less than or equal to 1.5 times the upper limit of normal. Cytokines in serum samples were obtained before dosing and 2, 6, and 24 hours after infusion for the first and third doses. Let's see. Complement factors were measured before dosing and 30 minutes, 2 hours, and 24 hours after infusion for the first and third doses. The pharmacokinetics use plasma and plasma filtrate. Unspecified time points were analyzed for total and encapsulated siRNA. A validated hybridization ELISA was used for each of the 2 siRNAs. ELISA is an enzyme-linked immune absorbent assay, or enzyme immunoassay. ELISA is a plate-based assay technique designed for detecting and quantifying substances such as peptides, proteins, antibodies, and hormones. So here's a little diagram. There are 3 major types of ELISAs. There is the indirect, which an antigen is added to the plate along with a blocking buffer. Suitable primary antibodies are added, and secondary antibody is added, which recognizes and binds to primary antibody. And then finally, TMB substrate is added and converted to detectable form. For the indirect method, so you apply a sample antigen to a surface, an enzyme-linked primary antibody is applied to the plate. After washes, only antibody antigen complete remains. A substrate is added, which is converted by the enzyme to elicit a chromogenic signal. And finally, the sandwich method. Let's say a plate is coated with suitable antibodies, a blocking buffer is added, sample is added to the plate, so antigen is bounded by capture and by a capture antibody. Biotin labeled detection antibodies added to the plate, and enzyme HRPO is added and binds to the biotin labeled detection antibody. Finally, TMB substrate is added and converted by HRPO to the colored product. Let's see, 3 separate core samples were obtained from the same tumor at each time point. 2 of the core samples were obtained for siRNA quantitation and for VEGF, KSP, qPCR, and 5' race. The 3rd core sample was sectioned and stained with H&E for histopathologic evaluation. Rapid amplification of cDNAs or RACE is a procedure for amplification of nucleic acid sequences from a messenger RNA template between a defined internal site and known sequences at either 3' or the 5' end of the mRNA. As described as a one-sided PCR, RACE results is the production of a cDNA copy of the RNA sequence of interest, produced through reverse transcription followed by PCR amplification of the cDNA copy. The only requirement for RACE cDNA amplification is that you know at least 23 to 28 nucleotides of sequence information or to design a gene-specific primer. Here's a diagram of the procedure for this experiment. And in order to perform the 5' race assay for this experiment, RNA adapter with known sequence was added to the 5' end of cleaved VEGF RNA, which was then reverse transcribed to cDNA synthesized by reverse transcription using cleaved VEGF RNA as a template, followed by PCR. The 230 base pair PCR product was cloned into a vector, which was subsequently sequenced to confirm cleavage site of VEGF by siRNA guided RISC.
Hello everybody, my name is Juliet and I will be presenting on the results of our research article. And the first part that I'm going to talk about is the results for the preclinical animal studies. And these were conducted on monkeys and rats. So in the liver was for the rats and in the spleens was for the monkey. So we observed for the monkey that there was an observed 50% weight reduction that was associated with the lymphoid atrophy in the monkeys. And those monkeys were treated with 4 doses of the 6 milligram per kilogram of the ALN-VSP. And that was given every 2 weeks. So this was not observed in the control animals that were treated with that same dose of the LNP-containing siRNA targeting luciferase. But on the target siRNA, the effect was observed. So this was instead of a non-specific effect of the LNP itself. So the lymphopenia reported in patients that received the small molecule kinesin spindle protein inhibitor. That meant that the KSP, instead of the VEGF inhibition, was causing the splenic changes in the monkeys. So now we move on to the mice. So the mice were implanted with the HEP3B human hepatocellular carcinoma cells. And that was so we could test this doses on them. So The first was a dose-dependent suppression that we observed of both the VEGF and the KSP mRNAs, and that concluded that there was a 50% reduction observed after 24 hours, with that dose being 4 milligrams per kilogram. So the generation of specific RNAi mediated VEGF and KSP mRNA cleavage products was measured using the 5' race assay, which we already talked about. And so with that, the pharmacodynamic changes expected with the KSP and VEGF inhibition were observed. And this was within 48 hours following that single dose of ALN-VSP. So that means that the tumor cells, they froze in the mitosis with the monoaster seen in all the liver tumors within all the treated mice. So this was not observed in control animals, again, just like the monkeys, not on the control animals, which is consistent with the KSP inhibition. So the mice that were given the repeated treatments, we observed a reduction in the tumor hemorrhage and the microvascular density. So this was comparable to animals treated with the anti-VEGF antibodies. So then after that, the anti-tumor activity of the ALN-VSP administered, those doses were given over 3 weeks, and we observed a 50% improvement in median survival relative to those control animals that were given the same dose. So the ALN-VSP has activity against the extrahepatic tumors, and this again was seen by the detection of the tumor cell monoasters after giving the treatment with the ALN-VSP and having the abdominal peritoneal HEP3B tumor implants in those mice. So now we get to move on into the in-human phase 1 and extension study results. So for those results, the patient with the endometrial cancer and the multiple hepatic metastasis, as well as the abdominal lymph node metastasis, was observed to have a disappearance of all target and non-target lesions. So this meant that there was a tumor regression that was observed and that occurred after the first 2 cycles of the doses of ALN-VSP. And so at the end of 6 cycles, that patient had a partial response with a complete regression of the lymph node metastasis. So this included a substantial shrinkage of the liver that was noted, and then a complete response was obtained after 40 doses. So the patient remained in remission and did complete the treatment after giving 50 doses over 26 months. So then after that, 2 other patients with RCC and extrahepatic sites of the disease, so that was lung, kidney, and lymph nodes, had stable diseases at all sites, so all sites meaning all of lung, kidney, and lymph nodes, for 8 to 12 months after receiving the VEGF pathway inhibition therapy. And so this was a really positive result. So another patient with the PNET and multiple liver metastasis was stable and had a stable disease observed within 18 months. So this was 36 doses that were given in the extension study. So after those 50 doses, there was 36 more doses that he continued receiving to have more of a research on him. So now we can move on to figure 1A. So before I explain this, the CT scans that showed the multi-liver metastasis were in the patients from the pre-treatment stage all the way up to the 40 doses of administration. So over here in the pre-treatment, you could see in the CT scan the tumors. So the arrows are pointing to those tumors. So in the pre-treatment, you can see them here, and you can see the 3 big ones here. There's one over here as well. You can also observe one here. There's no arrow, but it says that it's right here. So the patients were given 12 doses of the ALN-KSP, and you can definitely observe a reduction. This one is very faint, but it's right here, the big one that they had. This one was very reduced, you can tell that, and over here it's almost disappearing. So after 40 doses of the ALN-VSP, you can see that the tumor has completely regressed. Also in this bottom one, you can see that it totally regressed here, and you can't observe it anymore. So this was the complete response in the endometrial cancer that was observed in the patient that I spoke about over here. Also for this one, for the phase 1, the CT scans that were taken every 2 treatment cycles, those aided to see the eligibility of the patients that were to continue the trial after. So that would be for the extension study. And all the patients were pre-treated with chemo and anti-VEGF receptor agents. So the majority of the patients were both hepatic and extrahepatic tumor patients. So there was a total of 277 doses administered on phase 1 and the extension studies. And for all of the cases, the disease progressed after and prior to chemotherapy and the experimental therapy with the hedgehog inhibitor. So figure 1b is mainly to see the decrease in the blood flow in the liver tumors. So over here, the average K-trans, that's the blood flow change from the baseline, which was 0. So this over here just gives you the number of tumors in all the patients. So this patient had 3 tumors, this patient had 1, etc. So these are the doses given to the patients, and we can observe here that the patients that were treated with the ALN-VSP, which was 24 patients, those were able to have evaluable DCE MRI scans. And what a DCE MRI scan is, just a dynamic contrast enhanced MRI, which will help us in the next figure to see the blood flow in those organs. And with the decreased blood flow, that just means that the ALN-VSP dosage is working because, again, the blood flow is decreasing in those tumors, meaning it's not living. So in this figure, we see that the average peak change in the blood flow in K-trans for 1 or more liver tumors occurred during the first week of the dose. And among all the 28 patients across all dosage levels, 46% of these patients had a 40% average peak blood flow reduction. So as you can see right here. And that's figure 1b. So in this next figure, 1c, you are able to see 3 tumors in the right lobe of the liver, and this is an abdominal MRI. So these are the 3 tumors. So again, the K-trans is the blood flow. And as you can observe, because of the DCE MRI, the red in here is the blood flow. As the dose is increased, the blood flow in the tumors decreases. So as you can see a lot more red here. This is day 4 of the post-dose 1, which is the ALN KSP, which is 0.7 milligrams per kilogram of the organ. You can already see a decrease. And this is day 7 after post-dose 1. So the changes in the blood flow and the gadolinium concentration are observed to happen in parallel and seen within days of the first dose in large and small tumors. So 3 tumors, like I said, are shown in the abdominal MRI, and the blood flow decrease is seen in the tumors as the doses continued to be administered. So in the next figure, there was a decrease observed in the spleen volume in patients that were treated with the ALN-VSP, and this was measured in the abdominal CTs of 25 patients with pre- and post-treatment scans. So after 2 to 6 doses, there was an average reduction of 37% in patients that were dosed at 0.4 to 1.4 milligrams per kilogram of that organ. And there was no clear dose response, but there was a large effect after 12 or more doses. So that's why you can see an increase after those dosages. And again, this is the number of doses that each patient, which is the bars, is given. This is the percent change in the spleen volume, and then this is the dosage level that that patient was given. So again, there's an average 37% reduction in that spleen volume. So the spleen volume is very important because you could see that the tumors are decreasing. So in the next figure, which is table 3, about 10 patients' biopsies showed that they had varying amounts of viable tumor with necrosis fibrosis, and that the amount of KSP and or VEGF siRNA couldn't be determined, but what this table shows us is the drug levels that were given and the changes in those tumors. So for example, this patient number 7, was given a dosage of 0.4 milligrams per kilogram of the tumor type colorectal. So not only did this patient have colorectal cancer, but they also had liver. And the viable tumor was 17%, which means that the weight of the liver was 80% of it. And then 3% of that liver was had fibrosis or necrosis. So that determined the drug levels that were given, and also you could see the change in the target mRNA in the patient. So this one went down 20%. Again, because 10 of the patient's biopsies showed that there was varying amounts of the variable tumor, some of these results were not able to be shown. So they couldn't be determined. The amount of the KSP could not be determined with this table. So the difference between the VEGF and KSP siRNA concentrations weren't significant, but because 2 of the patients had biopsies that were 96% to 100% viable tumors, this suggested that the drug delivery to the tumor itself was positive. And because the biopsies from the other 10 patients, again, those couldn't be determined, but 2 of those you could. So among the post-treatment biopsies, 2 patients biopsied 48 hours after dosing, and those were the 2 that were 100% and 96%. And those were composed of predominantly of normal livers and had little to no viable tumor. So they had a substantial increase in the specific cleavage product for the VEGF and mRNA by sequencing that was compared with the basal level. So this is for figure 2A. So this shows the sequencing from the VEGF 5' race assay, and this is post 2-day treatment with the dose number 1 of ALN-VSP. So on here, the cyan bar, so this blue bar, that represents the cleavage product of the VEGF mRNA, so right here. And this is the positioning on the patients, on the sequence. And so the fragments that are shown in orange, so all of these, are the VEGF fragments. And so the p-values, so these p-values that you see here are the ones compared with the basal or pre-dose level that was derived from the t-test, as said before. And so this was of a liver, a normal liver, and the patient with the colorectal cancer metastasis as well. So again, this is 2 days after treatment with the dose number 1. So for figure 2B, this just showed that it was Agilent microfluidic bioanalyzer, and these results are from also the VEGF 5' race, and this was the second round PCR that was conducted on an untreated normal liver. Then it was conducted on a liver with tumor biopsies from 2 patients. So it was patient 16 and patient 17. So these arrows, this arrow, it points to the predicted specific VEGF mRNA cleavage product, which is roughly around 230 base pairs. And so again, this is in a normal liver, so pre-normal liver, and then you had pre-dosage, and then you had post-dosage. So you could see that it was predicted to be around here, and that's what we got in the results. So now we move on to figure 3. So figure 3A was the complement and cytokines that were seen in the patients that were treated with the ALN-VSP, and that was the BB complement. So this shows the peak fold change in the BB over the baseline, and this is the dose given. So the arrows show that there was acute infusion reaction, and the lines are the mean. So while there was an increase in the VB complement were seen in most patients targeted, those patients with the IRR at 0.4 to 1.5 milligrams per kilogram, so those patients tended to have more BB induction. And so additional cytokines were seen that also peaked at 6 hours and were normalized by 24. So after 24 hours, the post-dose included the IL-6. So that's what you could see here in figure 3B as well. So for figure 3B, again, it shows the same, but this one just doesn't have the IL-6 dose. This one has both. So the peak and the IL-6. That's why there's more. So the TNF-alpha cytokines were not induced, including the IL-1B and IFN, but being most commonly, they were associated with toll-like receptors, so TLRs, and that was the activation by a single or double-stranded RNA. So the symptoms that these patients got, which is what this figure shows us, is that they had typically occurred 4 to 8 hours after they were infused with the dose, and it was around the same time of the peak cytokine induction. So there was no cytokine induction in the patient with the endometrial cancer with the complete response treatment. And so the analysis of the plasma pharmacokinetics, you can see it in the next slide that we will show. But again, some of these patients showed the side effects such as chills, rigor, some with both chills and rigors, and it also showed the mean of those. So figure 4 is a little complicated, but you can see that they had the KSP siRNA, the dosing over here, and then over the time. So this is the analysis of the plasma pharmacokinetics for the VEGF and the KSP siRNAs. So that showed that the maximum concentration, which is the Cmax over here, and that is, and the area under the curve, so that's the AUC over here. And those were similar for both of the dose proportional. So as you could see, this is just KSP siRNAs. The second one is, the one on the right is the VEGF siRNAs. And you can see that they're very similar to each other. And again, this is also the time. So you could see how they're very similar and they're very proportional. So approximately 97% of the siRNA detected in the plasma was encapsulated. So this means that the pharmacokinetic profile in man was very similar to what was observed in non-human primates, so monkeys. And this was similar profiles after the first and third doses, and they had no evidence of accumulation. So the ALN VSP phase 1 pharmacokinetic data showed that the mean plasma pharmacokinetic results for KSP and VEGF showed that the levels following the first dose of ALN VSP reduced. So as they increased the dosage, you could see a reduction over time. So those are the results that we obtained from this research. And next you will hear the discussion and after the conclusion. Thank you.
Hi, I'm Tyler, and I'll be going over the discussion. The discussion basically talked about 4 different points, 4 different results. The first result was the safety of the IV doses, and what they found was that overall the ALN VSP was more tolerable than other cancer treatments. They also said that the infusion-related reactions that did occur with the treatment were likely more related to the LNP part of the treatment and not necessarily VEGF or KSP inhibition. And they think that because the infusion-related reactions weren't correlated to a cytokine induction. And then the second point was the ability to be measured in biopsies. So they were able to find both siRNAs in liver tumor biopsies and in metastases in the adrenal gland, lymph nodes, and abdominal cavity across multiple different tumor types. And so that's a promising result because it shows that the ALN VSP was successfully delivered to tumors. And they know it was successful because the samples of these biopsies were of pure viable tumors. However, they did find cleaved VEGF mRNAs in healthy kidney tissue, which does prove that the mechanism works, that it does cleave the VEGF mRNA. However, it shows that the treatment didn't only go to tumors. The same happened with KSP mRNAs. They were able to find cleaved KSP mRNAs, which is proof of a successful mechanism, but they also found them in the liver and other metastases outside of the liver. Okay, the third point is the pharmacodynamic effects in metastases. And they were able to see that with treatment, tumor blood flow decreased, as seen on the DCE MRI. And that was in nearly half of the patients, which in other studies has been shown to be an effect of anti-VEGF. However, they still would need to clarify or study the correlation better, because they weren't able to prove that it was that anti-VEGF response was actually correlated with the delivery of the ALN VSP. And likewise, they saw an anti-KSP response, which was a change in spleen volume. The fourth point of the discussion was the anti-tumor activity, which was another very promising result. They had a complete response, which was not expected for phase 1 of this experiment in trials. And many patients had a year to a year and a half prolonged disease stabilization, which all of these results are very promising for the ALN VSP treatment. And one way that they could tell that it was that the complete response in disease stabilization was due to ALN VSP was that there wasn't a cytokine induction during the treatment, which is related to would be an immune response. So they think that because there wasn't cytokine induction, the prolonged disease stabilization and the complete response weren't due to an immune response that would target the tumors. And then quote straight from the paper that I think sums up their findings and their promising findings is they say that subsequent phase 2 trials with ALN VSP focusing on a single tumor type, enrolling patients with fewer prior therapies and treating all patients at the same active dose level will be better suited for examining the relationship between target modulation and clinical response to therapy. So hopefully they can continue trials, phase 2 trials, and improve the treatment and correlations to side effects and correlations to the complete response and prolonged disease stabilization so that this treatment could help people.
In conclusion, the results prove the effectiveness of RNAi to helping people that are dealing with cancers that involve the liver. initial method of using RNAi and LNP formulated siRNAs, lipid nanoparticles have been proven to be effective in the delivery of siRNA to the liver and to tumors within animals. RNAi is used for the silencing of genes connected to diseases, targeting the genes of VEGF and KSP. The LNP formulated siRNAs are safe and tolerated to use. From the results of the animal experiments, the reduction of spleen weight in monkeys was associated with lipoid atrophy. To observe how LNN VSP worked, mice with tumors were treated to determine the anti-tumor effects of LN VSP. This helped to determine whether LNN VSP was safe enough to be used on humans. From the results from the in-human trials, all target and non-target lesion disappeared in patients with endometrial cancer and multiple hepatic metastasis, as well as abdominal lymph node metastasis. The blood flow is decreased due to liver tumors. Risks. You need to have a spleen and a full liver to undergo the treatment. The safety are, compared to other tumor treatments, it's more tolerable. Lower dosages of IV to treat IRR, measuring the ability of biopsies across multiple tumor types. siRNAs were detected in liver tumor biopsies and in metastases in adrenal gland, lymph nodes, and abdominal cavity would only be successful transfer of pure viable tumor biopsy samples to the tumor. The cleaving of VEGF mRNA and KSP mRNA also were used to measure the ability of biopsies. In summary, the results can show that from the data, the right patient candidate is likely to be successfully treated through this new form of treatment. The future works are the research shows safety, pharmacokinetics, RNA mechanism of action, and the clinical activity with first-class LNP formulated RNAi therapeutic patients with cancer. This will allow RNAi to be harnessed to promote specific multi-targeting while also increasing the number of druggable targets, which will help with further development of drugs in oncology.